Assalamualaikum. My name is Siti Nurashaira binti Muhammad Tarmizi. In this video, I want to talk about DNA replication. What is DNA replication? DNA replication is the process by which the genome's DNA is copied in cells. Before a cell divides, it must first copy or replicate its entire genome so that each resulting total cell ends up with its own complete genome. This diagram shows the DNA replication where the DNA was copied and divided. A cell must create 2 copies of its DNA to allot to each of its new daughter cells before it may divide. This process known as DNA replication and happens in 3 steps during the S phase of the cell cycle, which are initiation, elongation, and termination. For the initiation, the first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and result in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. For elongation, an enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made and the process is repeated again. And the last is for termination. Once the new DNA has been made, the enzyme isonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.